everyone. My name is Xueying, and I'm from University of Pittsburgh. Today, I'm very happy to present our work using the Wavelet transform to identify the spatial scale of multivariate activity patterns in fMRI data. So we all know that from individual neurons to larger functional brain regions, the brain is hierarchically organized on different spatial scales. So the brain region that we were interested in is occipital temporal lobe, which is a very important brain area for visual features processing and object recognition. However, the question of whether information in this brain region is represented on fine or coarse spatial scales during this visual processing remains debatable. For example, some studies have argued that visual features such as the orientation information is represented at the fine spatial scales. Others have also suggested that visual features decoding in primary visual cortex reflects a large-scale bias. Past studies have used a method to tackle this issue called spatial smoothing, in which they smooth the brain patterns before they do the classification. For example, if they want to study a finer scale, they might use a smaller kernel such as the 4 millimeter. And if they want to study a coarser scale, they probably would use a kernel, say 8 millimeter. However, those spatial scales are not independent with each other. In other words, the information extracted on the scale using the 4 millimeter kernel might be overlapping with the information using the 8 millimeter kernel. So how do we overcome those problems if we want to study the information represented on different spatial scales, which are more stringently defined? So the method we used in our study as a solution to this problem is called the Wavelet transform. It is a way of using a localized wavelet or a small wave-like oscillation to decompose the signal into a hierarchically independent spatial scales, and where the coefficients at a particular scale and location represents the response of the signal to the wavelet applied. It has advantages over the traditional Fourier methods for examining the spatial structures. So Fourier analysis depend on the sine and the cosine waves that's stretching out to infinity, which can lead to very poor performance in capturing local features of the signal, such as sharp specks. And in contrast, the wavelets are extended to a finite range. It is very suitable for studying the local features of the signal. We can see here that there's a small speck in the center of the figure. So those local features can be better captured using those wavelets. So in our study, we used the wavelets to investigate the spatial scale of neural representations for visual categories in occipital and ventral temporal cortex. Here's the experiment design. So the data was first analyzed in Coutange and Thompson-Shields paper published in 2012 about advantages of fMRI short runs over long runs, and the data made available to us by Thompson-Shields lab. So there were 8 participants, and they completed the fMRI scan as they viewed 4 different categories. We can see from this figure here, the 4 categories are faces, places, and man-made objects and fruits. So only the data from the 16 short runs were analyzed in our study. So each short run has 36 TRs and has 1 block of each category presented in a random order, and each block has 10 trials. So for more information about this experiment design or about the data of long runs, please refer back to their paper. Okay, so yeah, I like this figure a lot. So here's a demonstration of the wavelet decomposition. So based on the 3D fMRI data volume, we use the wavelets to calculate 5 unique scales that describing the coarseness. So here I'm using the conditional faces and the wavelet coefficients projected onto a 2D surface for 5 different spatial scales. So the first row here is the brain region of occipital, and the second row is VT. So the values in the parentheses indicate the number of coefficients transformed using the wavelet on each scale. So we can see that from this, as the scale gets larger, so the resolution gets coarser. So we have those new spatial dependent features that were generated using the wavelet, and then we pass those features to a XGBoost classifier to classify those 4 different categories, and we repeat it for 5 different spatial scales. And here are some interesting results. So in occipital cortex, we were able to classify those 4 visual categories at coarser spatial scales. And here we can see that at scale number 4 and number 5, which contain the low frequency information. I think those results can be taken as the indication that there are significant differences in the amount of information represented at low frequency scales for those categories, which is sufficient enough to drive the categories classification. However, in VT, we were unable to classify categories at any spatial scales based on those new features. And the significance of the results were validated using the 10,000 times permutation test. Okay, so hopefully I'm not overwhelming you with this a lot of information. So to sum up, what is the take-home message here? So based on the results, we can say that the amount of information do differ between the spatial scales, and is largely differentiable at larger spatial scales for different categories in occipital cortex. I think the main virtue of this wavelet method is that it not only can define the fine versus coarse spatial scales more stringently, which is very suitable for any spatial scale dependent analysis, but also it can look at the neural representations differences based on new measurement. Also, I think this study is one of the few attempts of using the wavelets to analyze the fMRI data. So I do believe that further exploration of the wavelets will definitely help us to get more insights and more interesting findings in these neuroimaging studies. Okay, so I guess that's it. So thank you for listening. I'm happy to take any questions you may have.
Fantastic. Thank you, Xueying. And in lieu of applause, because we can't do that in Zoom, if you enjoyed the talk, please drop some praise in the chat. Very nice. So we have a couple questions that came in from the panelists who unfortunately cannot use the Q&A box because that is how that works. Very quickly, the first question is, what was the train and test data split for your data?
So we use the 16-fold cross validation. So we have 16 runs. So each one will work as the training testing set for the classification.
Great. And then Mohammed, you had a question? You raised your hand?
Yeah, I was writing it, but yeah, I can ask it here also. So from what I understand, the finer scale, of course, has more data points or more features. So could it be that because of that, it's overfitting the classifier as opposed to the lower scale?
Yeah, since due to the time limits, I didn't point it out very specifically. Actually, we use because we for example, from the first scale, we have more coefficients, right? Actually, instead of using all the coefficients, we calculated the variance of the coefficients, and we call it as the global energy. It's we use this kind of feature. We are not throwing in all of the coefficients. We just use the variance of the coefficients. So yeah, so I think this feature can capture the variability in the brain signals. So that was used for the classification. Does that answer your question, Mahad?
Yeah, it's just that I'm not familiar with this.
Yeah.
With this analysis, so yeah, I'll have to go around and read about it.
Yeah, I feel like there's definitely still a lot unknown about this method. I think there's more exploration of this method definitely can help us to understand better of this method. Yeah.
We also have another question from Avital Hami in the audience. And so Avital is asking, I wonder if the low spatial scales would represent the anatomical differences between visual categories, different regions selected for different categories.
I think that's a very, very good point. So for our study, I think we are mainly interested in like the amount of information represented on different spatial scales. But I think that, yeah, that might be a possible like direction for future works, that the lower scales or the finer scales can represent like different anatomical differences. Yeah, I think that's a good point. But yeah, since we didn't do the study in that area, so I cannot like answer that very, very, very clearly. But I think that's a good like direction for future work.
Great, thanks. I have a question actually, because we have a couple minutes left before our next speaker. One of the things that really struck me, this is kind of a half comment, half question. One of the things that struck me is that the classification accuracy is not necessarily the only thing that's going to change as you change the spatial scale that you're looking at. Of course, there's going to be lots of different things, just the nature of the representation itself. So I wonder if you've thought about extensions of this or next steps that might involve something like representational similarity analysis or other kinds of ways of assessing the nature of the representation, the content of the representation across different regions in a way that speaks also to these different spatial scales.
Yeah, I think those are very, very good points that, so for, like I said, for our study, we only mentioned, like, we were only interested in the occipital cortex or like temporal, like ventral temporal cortex, because these two regions are very critical for visual processing. But I think it would be very interesting to like apply this method to other brain areas to study that, like how neural representations might differ. Like also I mentioned that in our study, we only use like try to examine the amount of information represented on different spatial scales. But I think for future work, it might be interesting to examine what kind of information, what exactly represented on different spatial scales, and what exactly the differences embedded in those information on different spatial scales. I think those are very, very good points that could lead to further investigation.
I think especially if you're trying to do cross-modality representational similarity analysis, like between EEG and MRI, that you might find very different answers depending on the spatial scale that you're looking in the fMRI pattern. So I think that would be really interesting to look at. And then we have a quick question also from Mohammed. Do you want to ask it yourself, or do you want me to ask it? Okay, Mohammed is asking, what kind of wavelet function did you use, and is there a reason for that choice?
I was hoping like someone would ask this question actually. The wavelet function that we choose for our study is called complex dt complex wavelet transform. So I'm not sure if maybe some of you may be familiar with wavelets, because wavelets have like different like families. So for our studies, we specifically use this dt complex wavelet transform for two reasons. First is shift invariant. Invariant means that the signals cannot be easily perturbed or disturbed by the motion. For example, I think this is especially good for dealing like neuroimaging data, because sometimes the motion cannot be fully corrected. So this method has these advantages for being signal shift invariant. The second reason for choosing this method is that it's directionality selective. For example, like for 3D volume, fMRI volume, there can be like 28 different orientations isolated to try to like look at the data from different orientations. I think it's very suitable for the 3D data volume. So that's why we chose this dt complex wavelet transform. Yeah.
Fantastic. Well, thank you, Xueying, for your great talk and for the fun discussion. Unfortunately, we need to move on now, or fortunately, we need to move on now. So thanks again. And our next speaker